Hello and welcome to Zoology 142 Online. Today's topic is the digestive system. So here are the student learning outcomes for today's lecture. After completing the lecture, you should be able to describe the major digestive processes. You should also be able to identify the major divisions of the digestive system. You should be able to identify the layers of the digestive tract and also discuss the structure and function of peritoneum. You're actually going to have to do some reading for this one because I'm not going to go over it in the lecture. And finally, you need to be able to identify the major structures in the mouth, including the tongue and the teeth, and discuss their role in mastication and digestion. Well, there's basically two reasons that we have to eat food. One is that we need energy. We need calories to help power our cells and provide us with the ATP necessary to run all the various cellular processes. The other thing that we need is raw materials. Our cells are made out of lipids, are made out of proteins, and they also have all kinds of other types of molecules in them as well. So we need raw materials to repair the cell membranes and to repair the proteins that make up muscle tissue. And we get these raw materials from the food that we eat. However, in order to obtain these raw materials and also obtain the energy from the food that we eat, we must first break that food down via a catabolic process called digestion. We have to break it down into small enough parts that these parts can then be absorbed through the digestive tract, which is mainly going to be in the intestine. So usually we're talking about taking a large polymer, let's say like a starch, and breaking it down into monomers like simple sugars. So they have to be broken down into the monomer stage before they can be absorbed. So we often talk about digestion as if it were one continuous process, when in truth it's about 5 or 6 different processes which are happening simultaneously. So the first of these digestive processes is something called mastication, and mastication is just a fancy name that means to chew. And so chewing helps to break our food down into smaller pieces. It doesn't actually divide, let's say, carbohydrates down into simple sugars, but it does help us to break a large, let's say, candy bar into smaller pieces, and this enables the enzymes in our digestive tract to more easily break down those smaller pieces, because smaller pieces have a proportionally greater surface area than larger pieces. So mechanical processing or mastication is an essential part of digestion. Okay, the next process is secretion. This is basically the production of things like stomach acid or digestive enzymes or even mucus by cells within the intestinal epithelium. And digestion proper is actually a process, a catabolic process, where we break down large polymers that are found in our food, such as carbohydrates, into the monomers. And this occurs because of the production of enzymes. We have enzymes in the mouth that help to break down starch. We have enzymes in the stomach that help to break down proteins. And finally, enzymes in the pancreas and also in the intestine that help to break down fats and other macromolecules. And so digestion is a necessary step in order for the next step to take place, which will be absorption. So absorption is the process by which nutrients are transported across the mucosal epithelium into the circulatory system. That is, either in the lymphatic capillaries or into the circulatory blood capillaries. And so absorption primarily occurs in the small intestine. So there's an answer to an exam question right there. The majority of nutrient absorption actually happens in the small intestine. And in order to absorb things, they tend to have to be in their smallest parts. So we have to break down proteins into amino acids. We have to break down carbohydrates into monosaccharides, and we have to break down fats into monoglycerides and also fatty acids. And finally, the last digestive process is elimination. This is where we get rid of any undigested material in the feces, and we're going to talk a lot about poop in this lecture. So now that we've had an overview of the major functions of the digestive system, let's take a look at the major anatomical divisions of the digestive system. The first of these is the gastrointestinal tract or GI tract. The GI tract is basically a hollow tube through which food and feces transit. The next major division are the accessory organs. These are organs that secrete something that go into the hollow tube, or they absorb some of the nutrients that come out of the hollow tube. So examples of GI tract organs include the mouth, pharynx, esophagus, stomach, small and large intestines, and also the rectum and anus. Whereas examples of accessory organs are things like the salivary glands, the liver, the gallbladder, and the pancreas. Remember, the accessory organs don't actually have food transit through them, but they are organs that produce something that goes into the digestive tract, such as enzymes, or they absorb nutrients from the digestive tract, for example, the liver. We're going to talk more about both GI organs and accessory organs in just a couple minutes. So let's take a look at the layers of the GI or alimentary tract. So starting from inside out, the first layer is the mucosal layer. The mucosal layer is the layer that lines the inside of the GI lumen. Remember, the lumen was just the name for the hollow space we find inside any hollow organ. So the mucosa in this case is composed of epithelial tissue that does a lot of the secretion and absorption. It also has a small lamina propria underneath this epithelial tissue. This is an area of loose areolar connective tissue that may also contain some circulatory capillaries as well as some lymphatic capillaries. And finally, the muscularis mucosae, which gives a little bit of three-dimensional structure to the mucosal layer. It's important to realize that the muscularis mucosae doesn't actually contract or generate any kind of significant muscular force. It only holds the mucosal layer in a nice three-dimensional shape so that secretion and absorption will be enhanced. So major functions of the mucosal layer include secretion, for example, secretion of enzymes or stomach acids. Another function is absorption. For example, the small intestine is an area of the body where we absorb a lot of the nutrients that we eat, the carbohydrates, the proteins, etc. All of these are absorbed in the small intestine. That is, they cross the intestinal wall and enter the bloodstream. The other function of the mucosal layer is to produce mucus, as the name implies, and the function of this mucus is to, in part, reduce friction. As food moves through the GI tract, it tends to abrade the GI layers, so mucus is one way to keep that abrasion from happening. And finally, the last function of the mucosal layer is to protect the underlying layers from digestion. For example, think about the stomach. How is the stomach different than any other GI organ? Well, if you said that the stomach is pretty acidic, that is, it contains acid, you're correct. The stomach has a pH of around 2 to 3, and that's a very, very low acidic pH. And so one of the functions of the mucosal layer in the stomach is to protect the underlying layers from being digested. And so the mucosal layer tends to have a lot of mucus in there, and this mucus is fairly alkaline, and it's there to help buffer any type of stomach acid. But that being said, it's important to realize that the mucosal layer is probably the layer that receives the most damage in the digestive tract. And on average, our mucosal layer, especially within our stomach, is going to be replaced at an amazing rate. On average, most mucosal cells are going to be replaced every 5 to 7 days. So that's an extremely high rate of turnover. And it's because a lot of the cells are being lost to friction, but also to acid and enzymes and other things like that. So underneath the mucosal layer, we're going to find something called the submucosa. And the submucosa is made up of moderately dense areolar connective tissue. The submucosal layer contains both blood and lymphatic vessels, as well as nerves. In fact, it's important to know that in fact you have more nerves or more nervous tissue within your digestive tract than you do within your entire spinal cord. So when people said, "Do you think with your gut?" you have to say, "Yes, I absolutely do. That's because I have a lot of neurons in my gut." Of course, we're not conscious down there, but there are a lot of unconscious or automatic decisions that are made in the gut for us by our enteric nervous system, which resides within the submucosal layer. So because the submucosal layer contains lots of nervous tissue, one of its functions is going to be to regulate the muscular contraction of the digestive tract, because the submucosal layer, as it were, is where the sort of brain is within the GI system. Another function is to transmit nutrients via large blood vessels and lymphatic capillaries. Remember that all our large blood vessels and lymphatic vessels are located within our submucosal layer. And finally, the last function of the submucosa is to innervate various secretory cells within the mucosal layer. Remember, innervate is just a fancy word which means to supply nerve impulses to. And so the submucosal layer is basically in control of acid secretion and enzyme production, basically anything that is controlled by nervous impulses. Okay, the next layer outside the submucosal layer is the muscularis layer. This layer can be composed of either skeletal muscle or smooth muscle, depending on where we are in the digestive tract. For example, in the back of the pharynx, we have a lot of skeletal muscle, so it's a voluntary area, whereas in most of the digestive tract, for example, the esophagus, the stomach, the small and large intestines, are composed exclusively of smooth muscle, and so they're involuntary. Now, we might have several different types of fibers. We might have circular fibers as well as longitudinal fibers. And this different arrangement of fibers helps us to segment food and helps with mechanical digestion, and also helps to propel the food through the digestive tract through a process called peristalsis. So major functions of the muscularis layer. One is the churning and mixing of food with enzymes. And another important function of the muscularis layer is that it regulates the passage of food from the mouth through the esophagus and eventually to the anus. So it's important for the directional movement of food from start to finish through the digestive tract. So given that we have smooth muscle in some parts of our GI tract and skeletal muscle in others, what parts of the digestive process are actually voluntary? Well, if you think about it, there are two voluntary processes going on in the digestive tract. The first of these is the act of chewing and then swallowing. These are both voluntary processes that are accomplished via skeletal muscle in the mouth and also the back of the pharynx. But once that food is swallowed, that food will move through the esophagus, the stomach, the small and large intestines through smooth muscle contractions, which are involuntary. And so the next voluntary movement will actually be the movement of defecation, that is, clearing the digestive tract of any undigested material through defecation. Okay, the final layer surrounding the GI tract is something called serosa. The serosa is made up of a serous membrane of areolar connective tissue, and it covers and surrounds the other three layers of the GI tract. Another function of the serosa is that it attaches the GI tract to the body wall, that is, it's confluent with the visceral peritoneum. Now, if you want to know what peritoneum is, it's that glossy, translucent tissue that covers the inside of a body cavity. Maybe you haven't looked at too many body cavities, but you've probably cleaned a fish once in your life, or maybe looked inside of an animal, let's say if it was in surgery, and you'll see this glossy tissue that covers the inside of the body cavity, and that is peritoneum. And so peritoneum and serosa are connected to one another. And in particular, the serosa also is a pathway for blood vessels and lymphatic vessels and for nerves to exit the GI tract and enter the main part of the body wall. Okay, now that we've looked at the layers of the GI tract, let's start at the very beginning. The process of digestion, in fact, begins in the mouth. And so the mouth has various structures that are going to help with digestion. The first of these are the lips, or labia. The lips really aren't important for breaking down food, but they can help us to grasp food, and they're also important in speech. Next, we have something called the hard palate. The hard palate is that bony area just in back of your incisor teeth, and it's the area where you have bony ridges, and it's an area where the maxillary bone and the palatine bones meet up. So your hard palate is hard. In back of that is something called the soft palate. The soft palate lies just below the nasopharynx, and it's the area where you would thrust your finger if you were ever trying to gag yourself. And finally, in the very back of the throat, we have something called the uvula. The uvula is a punching bag-like structure which helps to cover up the nasopharynx when we're swallowing liquids or foods. And what this does is it helps to prevent us from aspirating fluid into the nasopharynx. But sometimes it doesn't work. If you've ever seen somebody that's laughing while they're trying to, let's say, drink a carton of milk, and all of a sudden you see that milk come out their nose, this is because their uvula hasn't been doing the job at blocking off the nasopharynx and preventing fluids from moving up there. So within the mouth we have several salivary glands which produce saliva. Now these salivary glands can be grouped into what we call minor salivary glands and major salivary glands. You really only need to know the major salivary glands. These include the parotid, the submandibular, and sublingual. Now let's take a look at the secretions of the salivary glands. The first of these is something called salivary amylase. Remember, the ASE ending on the end of a word means that it's probably going to be an enzyme. And so amylase is an enzyme that helps to break down amylose, or starch. Other secretions of the salivary glands include something called mucin. Mucin is a mucus-like protein that holds food particles together so they can be more easily swallowed. A minor component of saliva includes something called lingual lipase. A lipase is an enzyme that helps to break down fats, and lingual lipase is something produced by the lingual salivary glands. This is a lipase that is inactive when secreted in the mouth and does not become fully activated until it reaches the stomach. And finally, the last secretion of the salivary glands is something called lysozyme and immunoglobulin A. These are antimicrobial proteins that are there to ward off infection and prevent bacteria from basically moving from our mucosal layer into the underlying layers and infecting the entire digestive system. So while we're on the topic of the salivary glands, we need to talk about a disorder called mumps. Mumps is caused by a viral infection of the parotid salivary gland, and it results in swelling of the parotid gland and also malaise or tiredness and also some trouble swallowing. And in some rare instances in pubescent boys, it can also lead to a tremendous swelling of the testes, which can result in sterility and probably quite a bit of pain. And so I remember being in grade school with a kid that had mumps, and a letter was sent home, and mom said, "Oh gosh, somebody's got mumps in your school." And I'm like, "Yeah, big deal. What does that happen?" And then she told me, and of course we all walked around and stayed very, very far from this kid that had mumps until he fully recovered. So another structure that's obviously important for mastication and digestion is the tongue. The tongue is skeletal muscle, so it's voluntary, and we use it to help manipulate food, to pack it in the back of the pharynx so it can be more easily swallowed, but it's also essential for speech, and it also has on there several papillae, or bumps, that have taste buds on there. And remember, the taste buds are there to help test our food to determine whether or not it's palatable, whether it's poisonous, and so it's important in tasting the food, and also, if that food is tasty, packing it in the back of the pharynx so it can be more easily swallowed. And of course, we couldn't complete any discussion about the mouth without talking about the teeth. The teeth are tremendously important in that they help us to grind up or masticate the food that we eat into smaller pieces that can be more easily passed through the GI tract and digested and absorbed. And so the gross anatomy of the teeth includes the crown, which is the visible part, as well as the root, which is the part which is below the gum line. Now let's take a look at the composition of the teeth. The inside of the tooth is made of something called dentin, which is about 70% calcium. And this is the type of calcium that's fairly hard, but bacteria can break through it fairly easily. And so if the bacteria, let's say, were to dissolve the overlying layer, the enamel, and it gets to the dentin, it tends to degrade very, very quickly. In this case, you need to have the cavity filled, otherwise you could have basically that rotten decay getting all the way down to the pulp cavity. So the layer over top of the dentin is the enamel layer, and the enamel layer is made up of 95% calcium phosphate, and calcium phosphate is a much harder version of calcium carbonate. It's fairly impervious. It's hard for bacteria to break it down or burrow through there, but it does happen, as anybody who's ever had cavities can tell you. But it tends to be much more resistant to corrosion than the underlying dentin layer. And then in the central part of the tooth, we have something called the pulp cavity. The pulp cavity contains the nerves as well as the blood vessels. If you have a dental carie or cavity get down to the pulp cavity, you will be in some tremendous pain, and chances are, unless you're able to patch that cavity very quickly, you're going to have to get a root canal, in which case we actually pull out all of the nerve, all of the pulp, and all of the blood vessels, and basically leave behind a dead tooth that has no sensation in it. The next layer is the periodontal ligament. The periodontal ligament is made up of fibrous connective tissue bands, which helps to connect the root of the tooth to the alveolar bone surrounding it. And this is very important to prevent the tooth from moving. And what we do tend to see is that people that have gum disease, advanced periodontal disease, the periodontal ligament actually begins to break down due to the widespread infection that's happening in the teeth and below the teeth. And as the periodontal ligament breaks down, the tooth will become more and more mobile. And so tooth mobility is generally a bad sign. If the dentist can wiggle your tooth, it means that periodontal ligament has been broken down through white blood cell action and also the action of bacteria. And so it indicates advanced gum disease. Now, most of you are probably aware that we have two different sets of teeth, and the first set of teeth that comes in are going to be our deciduous teeth. So deciduous means to fall away, and the deciduous teeth consist of 20 teeth that are going to be our baby teeth, and these are eventually going to be lost as our permanent teeth come in. And so we tend to have around 32 permanent teeth. The teeth can be divided into groups, and the incisors are the teeth that are up front in your upper and lower jaw. The incisors have sharp cutting edges which help to cut food. For example, it helps us to tear steak if you're eating steak, or bite off chunks of chicken. So incisors are important for cutting and also prehension or grasping of food. The next group of teeth are the cuspids or bicuspids, which again are sharp teeth which help to cut and shred food, whereas the premolars and molars have flatter surfaces and they're primarily used for crushing and grinding food. So think about the types of food you tend to chew at the back of your mouth. These are things like hard nuts that are very difficult to break down, and it's more easy to use the flat occlusal surfaces of your molars and premolars to grind these foods up. And so if you ever watch, for example, cows chewing their cud, you'll see that they have this nice side-to-side motion with their jaws, and this side-to-side motion helps to break down the vegetable matter that they're eating using their large flat molar teeth. So just a summary of what's going on in the mouth. Remember, the major process here is mastication, and mastication happens because we have these nice sharp teeth that are used to chew and break down our food. In addition, we have salivary secretions, which are, for example, helping us to start carbohydrate digestion. Remember that salivary amylase helped us to digest carbohydrate. And finally, we also have the tongue in the mouth, and the purpose of the tongue is to push the food back in the pharynx, and it's also used to test the food to see if it's palatable, and also used in speech. So once we chew that food up, we're going to use our tongue to move that food back into the oropharynx. Remember that the pharynx was a muscular area of the digestive tract that extends from the internal nares to the esophagus. Initially, it's going to be lined with skeletal muscle, but then after we swallow and it goes down the esophagus, we're going to be talking about smooth muscle. And the lower part of the pharynx is the junction between two body tracts, that is, the digestive system and also the respiratory system. And the mouth and pharynx are the areas where deglutition or swallowing are initiated. And there are 3 different stages to swallowing. And the first of these stages is called the voluntary stage. This is a process where we're pushing that bolus of food into the oropharynx using the tongue. Remember, the tongue is skeletal muscle, and that skeletal muscle is under voluntary control. Hence, this is why we call this the voluntary stage. The next phase is the pharyngeal phase. This is when the respiratory tract is temporarily closed as the epiglottis covers over the glottis. And the epiglottis helps to divert the food away from the trachea into the esophagus. So the pharyngeal phase is involuntary. And finally, after the pharyngeal phase, we will now enter the esophageal phase. This is the period where we have muscular contraction behind the food and relaxation in front of the food, and this movement helps to propel food from the esophagus into the stomach. And just like the pharyngeal stage, the esophageal stage is involuntary because it involves smooth muscle. And so here we see an example of the voluntary phase. This person has chewed up or masticated some food, and they're now packing it in the back of their pharynx using their muscular tongue. And remember, tongue is skeletal muscle, so it's voluntary. At this point, the food is in maybe the oropharynx. It hasn't entered the main part of the pharynx, and it hasn't passed across the epiglottis. So right now, the epiglottis is open, allowing both breathing and chewing to happen. Now, once we enter the pharyngeal stage, that food will move across the epiglottis, causing the epiglottis to close, and so during this period, we can't actually breathe. The food is moving over the epiglottis, and the epiglottis is diverting it into the esophagus rather than having it go down the trachea. And so this is a period in time where if we have food get stuck, we can actually have somebody suffocate because the respiratory tract is closed up by the epiglottis. So while we're on the subject of digestion, we should talk about something called the sphincter. A sphincter is a ring of muscle tissue, either smooth muscle or skeletal muscle, that surrounds a hollow tube and restricts the passage of materials through that tube. For example, we have an upper esophageal sphincter, and this is a smooth muscle ring that allows food to pass from the pharynx into the esophagus. And once that food moves into the esophagus, that upper esophageal sphincter will then contract, and that helps in part to prevent things from moving back the other direction. So again, once we swallow that food, the process will cease to be voluntary and will now become involuntary. And so the next involuntary stage that we're going to cover is something called peristalsis. And peristalsis happens during the esophageal phase, where wave-like contractions of smooth muscle contract behind the food and cause relaxation in front of the food, which eventually basically squeezes that food into the stomach. So now we've chewed up that food and we've swallowed it, and the food has moved into the stomach. And so the stomach is probably one of the best known digestive organs, but can you tell me what its function is? One of the primary functions of the stomach is to serve as a food reservoir. Basically, it allows us to eat a large quantity of food and break down that food at our leisure. So think about the last time you went to an all-you-can-eat buffet. How much food were you actually able to pack away? For some of us, it can be quite a bit, and that's because the stomach has this amazing ability to expand under increasing pressure. And so one main function of the stomach is to serve as a food reservoir or storage tank. Other functions of the stomach include both secretion and digestion. The stomach secretes several different substances, including hydrochloric acid, enzymes, and also mucus. And these help to both break down our food products and also help them to move more easily through the digestive tract. Another important function of the stomach is mixing. The stomach is basically a muscular bag which contains lots of enzymes, and the muscles and the muscularis layer help to mix these enzymes with the semi-digested food particles, and this helps to enhance digestion. We call this semi-liquid food that is present within the stomach and other regions of the GI tract chyme. So the fourth and very important function of the stomach is to regulate the delivery of this semi-digested chyme to the small intestine. The small intestine's major function is simply absorption of nutrients along with continued digestion. However, it can only accommodate a little bit of food at one time, and so the pyloric sphincter or pylorus is an important valve that helps to regulate the movement of food from the stomach into the first part of the small intestine, which is called the duodenum. So we said so far that the stomach is important for mechanical processing, that is mixing, it's important for acid secretion, for enzyme secretion, but is it important for absorption? Well, the answer is actually no. The stomach is actually a very poor absorptive surface. That's because the only thing it really breaks down is proteins. But the other thing we should know about the stomach is that it has a very thick layer of alkaline mucus lining it. And this alkaline mucus helps to protect the underlying mucosal cells, but it also prevents absorption from being very efficient. And so as a result, most macromolecules are not absorbed across the stomach mucosa, but they will be absorbed across the intestinal mucosa. So now that we've learned a little bit about the function of the stomach, let's go take a look at the major anatomical divisions of the stomach. The most superior portion of the stomach is called the cardia. The cardia lies just inferior to the diaphragm, and the cardia is the area where the esophagus and the stomach connect. And at that junction there will be something called the cardiac sphincter. Remember, a sphincter is a layer of muscle tissue that helps to regulate the passage of substances from one tube to the other. In this case, the cardia prevents the reflux of, let's say, stomach acid from the stomach into the esophagus. Just inferior to the cardia is something called the fundus. The fundus is a large rounded area of the stomach, and this is an area that is important for food storage as it has very few muscular contractions happening here. So if you eat a lot of food, let's say Thanksgiving dinner, this is where you're going to store most of that food until it's ready to be digested. The next major region of the stomach is the body. The body is the mid part of the stomach, and it's an area where we have quite a lot of muscular contractions. So it's an area where there's a lot of digestion going on. Any food being actively digested, chances are is going to be within the body. And finally, the pylorus is the junction between the duodenum, or first loop of the small intestine, and the posterior part of the stomach. The pylorus is a muscular valve that has a very small opening in it, and this opening helps to regulate the delivery of chyme into the small intestine. Because unlike the stomach, which can deal with large quantities of food very quickly, the intestine can only deal with a little bit of semi-digested chyme at a time, and so the pyloric valve tends to be very narrow in diameter, sort of like the nozzle to a Tabasco sauce bottle. If you've ever shaken a Tabasco sauce bottle, you know that no matter how hard you shake it, only a little bit of sauce comes out each time, and that's the same for the pyloric valve. Each time the stomach contracts, only a few milliliters of chyme move from the stomach into the small intestine. And so here you can see a sagittal section of the stomach. Up top you can see the esophageal sphincter or cardiac sphincter. Again, this is a smooth muscle band that prevents, for example, stomach acids from moving from the stomach up into the esophagus. Below that you see rugae. Rugae are basically pleats that happen within the stomach. And these pleats allow the stomach to expand a little bit when we eat large quantities of food. They're also important marginally for increasing the surface area for secretion of enzymes. In the left-hand side of the stomach, you can see the pyloric sphincter. Remember, this is a narrow region that helps to regulate the delivery of chyme from the stomach into the small intestine. And finally, the duodenum was the first loop of the small intestine, just posterior to that pyloric sphincter. Hello and welcome back to Zoology 142 Online. This is part 2 of our lecture on the digestive system. So in today's lecture, we're going to complete the review of the external anatomy of the stomach. We're also going to describe the two types of digestive processes, that is, mechanical digestion and chemical digestion, that happen within the GI tract. We're also going to describe the effects of parasympathetic nerve impulses on gut motility. After you complete today's lecture, you should understand the three phases of gastric digestion, and you should also be able to describe which factors promote and inhibit gastric emptying. And finally, you should be able to describe the digestive functions of the pancreas. We're going to leave the gallbladder and liver for part 3 of the lecture on the digestive system. So if you remember back to the first lecture on digestion, we said there's actually two types of digestion going on inside the GI tract. The first of these is mechanical digestion. This happens when a muscular organ macerates the food and mixes it with the digestive enzymes. And this type of mechanical digestion happens within the stomach, and it also happens to some degree within the small and large intestines. We also have chemical digestion going on inside the stomach. We have, for example, acid that's being produced, as well as enzymes that are being produced. The main enzyme being produced inside the stomach is the enzyme pepsin, and pepsin, if you remember, digests proteins. So when food first enters the stomach, there will be a mechanical process of mixing waves generated by periodic contractions of the smooth muscle within the wall of the stomach. And these contractions happen every 15 to 30 seconds. They tend to be least intense in the fundus, where the food is stored, and most intense in the body of the stomach. With each of these contractions, a small amount of chyme, which was semi-digested food matter, is propelled through the pyloric sphincter into the duodenum, which is the first loop of the small intestine. So this drawing shows a little bit of the histology or tissue structure of the wall of the stomach. Just like any other GI organ, the mucosal layer is composed primarily of epithelial tissue. In this case, we're talking about simple columnar epithelium, which is really good for secretion. Remember, this simple columnar epithelium is also covered by a very thick alkaline mucus layer that helps to protect it from digestion. You can see that there are grooves or rugae in between the individual folds in the stomach lining. And again, this helps to increase surface area, which helps to promote secretion of enzymes. Below that, you can see the lamina propria, and the lamina propria is where we have some areolar connective tissue, along with some capillaries and also some lymphatic capillaries. Below that, we see our submucosal layer, which contains the majority of the larger blood vessels, larger lymphatic vessels, and also the nervous tissue. Remember, the GI tract contains a lot of nervous tissue that helps to control contraction and movement of food throughout the GI tract. And finally, we have 2 to 3 different layers that comprise the muscularis layer. The muscularis layer is made up of smooth muscle, so it's involuntary, particularly when we get down to the stomach. So these contractions that are generated every 15 to 30 seconds are caused by nerve impulses, and these nerve impulses stimulate contraction within the muscularis layer. So now that we've had an overview of the histology of the stomach, let's go back and take a look at the mucosal layer, because this is where the action is. This is where secretion is going on. So we're going to talk about the different types of cells that secrete things like mucus, enzymes, and acid. I should point out that most of these cells are in fact exocrine cells. That is, they have a duct or passageway for excreting a fluid or enzyme into a body cavity. And so the first of these cells is something called a mucous neck cell. As the name implies, it produces a lot of mucus. And this mucus is integral or essential for protecting the underlying layers from digestion. It's a very thick mucus layer and is also somewhat alkaline, which helps to buffer or resist the acidity generated inside the stomach. The next cell type is something called the parietal cell. The parietal cell is responsible for secreting something called intrinsic factor, which we'll talk about later, and also hydrochloric acid. Hydrochloric acid is essential for the deamination or denaturation of proteins. It doesn't actually chop proteins into pieces, but it does help it to unravel so that our protein digesting enzymes can work more efficiently. Also found within the mucosa of the stomach are the chief cells. Chief cells secrete the enzyme pepsin, but they do this in an inactive form called pepsinogen. Remember that pepsin is going to be responsible for the breakdown or catabolism of proteins into amino acids. Another function of the chief cells is to secrete a little bit of gastric lipase. Gastric lipase will be responsible in part for breaking down fats, but the gastric lipase doesn't tend to be very efficient. We tend to have most of our fat digestion, in fact, going on inside the small intestine and due to secretions of the pancreas rather than of the stomach. So the big picture here is that the stomach primarily digests proteins. It doesn't do very good digestion of either fats or carbohydrates. Another type of cell found within the mucosal layer of the stomach are the G cells. G cells, in fact, are not exocrine cells, they are endocrine cells. They produce a substance called gastrin, and gastrin helps to set the timing for stomach contraction and also the secretion of various substances from the stomach, for example, acid, mucus, and also enzymes. Now that we've had a brief overview of what each of these cells does, we're going to look in detail and talk about how they secrete their secretory products. So the first of these is the parietal cells, and if you remember, the parietal cells secrete hydrochloric acid. In order to understand how hydrochloric acid is made, it's essential that you understand the diagram at the left side of the screen. So the first process of hydrochloric acid secretion is in fact the secretion of something called carbonic acid. Carbonic acid is formed by the carbonic anhydrase enzyme. Basically, we take carbon dioxide, which is plentiful inside the blood, and we mix it with water to make carbonic acid. This carbonic acid will then dissociate into hydrogen ions and also bicarbonate ions. So remember that hydrogen ions are acidic and bicarbonate ions are slightly basic. And so if we left these two together, they would somewhat buffer each other. So to prevent this buffering process from occurring, what happens is the parietal cell will actually secrete that bicarbonate ion into the bloodstream while it places the hydrogen ion directly into the stomach lumen. So the hydrogen ions will help to reduce the pH of the stomach lumen, while at the same time the bicarbonate ions are actually raising the pH of the blood. And we call this process the alkaline tide. That is, after a large meal in which we need to produce a lot of acid, we also secrete a lot of bicarbonate into the bloodstream, and this tends to raise the pH slightly. This usually doesn't cause a problem except if somebody's been throwing up again and again and again. For example, think about somebody that has bulimia and they've been binging and purging. If they throw up several times a day, their stomach is having to generate more and more stomach acid to replace what was lost when they vomited. As a result, they're also generating a fair amount of bicarbonate ion, which is also being excreted into the bloodstream. And this can in fact lead to something called alkalosis, which is a higher than normal pH of the bloodstream. If extreme, this alkalosis can in fact be life-threatening. So now that we've talked about acid secretion by the parietal cells, let's go on to talk about pepsin secretion by the chief cells. Remember that pepsin was an enzyme that's going to help us to break down polypeptides or proteins into individual amino acids. So as it turns out, pepsin is actually secreted in an inactive form called pepsinogen. Now you might be asking yourself, why do we want to synthesize an enzyme that's inactive? Well, think a little bit about what that enzyme does. Pepsin is an enzyme that helps to digest proteins. Now think about what the mucosal cell layer is made out of. Well, it's made out of phospholipids and a lot of proteins. And so we don't want our cellular proteins to be broken down by this enzyme. So we initially secrete it in an inactive form called pepsinogen. Once the inactive pepsinogen is excreted into the stomach lumen, it will then come in contact with all the hydrochloric acid that was generated by the parietal cells. The hydrochloric acid is responsible for activating pepsin. That is, it helps to convert pepsinogen, which is inactive, into pepsin, which is the active form of this protein-digesting enzyme. Once activated, pepsin can readily aid in the catabolic process that helps to break down proteins or polypeptides into dipeptides and single amino acids. We're now going to take a look at the phases of gastric digestion. Remember that the word gastric means stomach, but the stomach doesn't decide when it wants to digest things. It makes sure that it gets input and stimulus from other areas of the body. So there's three phases that we're going to talk about: the cephalic phase, the gastric phase, and also the intestinal phase. So the first phase of gastric digestion is in fact the cephalic phase. If you remember back to medical terminology, cephalic means head. And this is important because the sensory information that's coming in here now is coming in through the special senses in the head. That is, we're smelling the food, we're maybe even tasting the food, but the food's not yet in the stomach. So this is a way for the nervous system to get the stomach ready for impending food, for food that's going to be coming down shortly. And so if you're smelling food, if you're starting to taste food, it's going to cause salivation, it's going to cause increased gastric motility, and it's also going to cause secretion of gastrin. Gastrin is a hormone which is going to stimulate secretion of hydrochloric acid and also pepsin or pepsinogen, and it's also going to increase gastric motility. That is the muscular contraction of the stomach. So I said the cephalic phase involves sensory input coming from the nose, coming from the eyes, and also coming from the taste buds. And this sensory input is routed to the brain, the central nervous system, and is also routed to the stomach via parasympathetic nerve impulses. So I'm imagining you probably don't remember what parasympathetic nervous system was, and so we're going to have a little bit of a review. So basically, there's something called the central nervous system, which is the brain and spinal cord, where all the processing happens, and there's also the peripheral nervous system. The peripheral nervous system is divided into a sensory division, which brings information to the brain, and also a motor division, which carries information away from the brain. And this motor division is subdivided into a somatic division and also an autonomic division. The somatic motor division deals with a voluntary muscular contraction. That is the contraction of skeletal muscle. So when you tell your arm to contract, it contracts, your elbow flexes or extends, and this is part of the somatic motor division. On the other hand, not everything controlled by the brain is voluntarily controlled, and everything that is automatic we call the autonomic nervous system. And the autonomic nervous system controls things like glandular secretion, it controls our heart rate, and several other automatic tasks within the body. And the autonomic system is subdivided into the sympathetic and parasympathetic divisions. These are antagonistic divisions. For example, the sympathetic nervous system will increase heart rate, whereas the parasympathetic nervous system will decrease heart rate. So that the mnemonics that we use to remember what these two opposing divisions of the autonomic nervous system do is rest and digest for parasympathetic and fight or flight for sympathetic. So the parasympathetic division is going to be essential for maintaining movement of food throughout the digestive tract. If you're very relaxed and you've had a big meal, that meal should digest fairly quickly. Whereas if you're exercising immediately after a meal, chances are that meal will not digest very well. You'll probably get cramps and you may even throw up. This is the reason why your mother always told you not to go swimming right after you have a big meal. That's because you're trying to digest and also exert yourself at the same time, and those tend to be two incompatible tasks. Finally, I should point out that some textbooks, yours included, also add another division to the peripheral nervous system, and they call this the enteric nervous system. That is, they split out everything that's in the gut and set it as its own division. I'm not going to do that in this course. In this course, I'm going to follow previous textbooks and basically say that the stomach and other GI organs are controlled by the parasympathetic nervous system. So now that we've had a review of the function of the autonomic nervous system, we're going to go back and talk about the second phase of gastric digestion, and this is called the gastric phase. So the gastric phase starts when food enters the stomach. If you remember, the stomach is pretty acidic because of all the acid generated by our parietal cells. Now what happens to this acid when the food comes in? Well, it becomes diluted, and this causes the pH to rise. The other thing that happens is that as we eat large quantities of food, the stomach is stretched, and this stretch is detected by stretch receptors within the wall of the stomach. So both the change in pH as well as the stretch of the stomach are detected by the enteric nervous system. With this sensory information in hand, the enteric nervous system will generate motor impulses which will stimulate gastrin secretion by our G cells. Remember, gastrin is going to stimulate secretion of other products within the stomach, for example, pepsin, mucus, and also hydrochloric acid. So this increased gastrin secretion will result in increased peristalsis, or muscular movement of the stomach, increased mucus production, and also increased acid secretion. So as more acid is added, the pH will then drop again down to set point levels. Once the acid concentration, or pH, reaches the set point, gastrin secretion will be inhibited. Remember that gastrin also causes increased peristalsis, which will cause increased emptying rates. That is, it will cause the stomach to empty faster. Now, once enough of the stomach volume has been emptied in the small intestine, the stretch receptors in the stomach wall will no longer be stimulated, and as a result we will shut down or reduce gastrin secretion. And so this will bring us back to set point. Our acid is now back at the normal levels. We have a pH of 2 to 3 in the stomach, and the motility is dropped down to normal set point levels. So only when food is present in the stomach do we have secretion of lots of gastrin, which results in a greater secretion of acid and also a greater secretion of pepsin, mucus, and greater stomach motility. So this is a negative feedback system. Now it's important to point out at this point that all the nerve impulses I've been talking about thus far have been basically equivalent to parasympathetic nerve impulses. These are things that promote gastric emptying and also promote digestion. If, on the other hand, we have sympathetic nerve impulses, this will in fact inhibit digestion and inhibit gastric emptying. So let's say that you're being chased by a tiger. Well, if you're being chased by a tiger, you're going to be in the fight or flight response. You're going to have high levels of sympathetic nerve impulses, and this is going to basically shut down digestion, shut down secretion of enzymes, and shut down acid production within the stomach. And so, as I was saying before, intense exercise and digestion tend to be processes that can occur at the same time. That is, they are mutually exclusive. So once food enters the duodenum, or first loop of the small intestine, we say that we've entered the intestinal phase of gastric digestion. And in the intestinal phase, we basically will slow down gastric digestion based on what's coming down into the intestine. And this slowdown process prevents the intestine, which can only deal with small amounts of food, from being overloaded too quickly. So in a nutshell, this is how it works. Fatty acids and glucose in the chyme cause the intestinal mucosa to release two hormones. The first of these is secretin. Secretin is made by the mucosal cells of the intestine, but it has its effect on the stomach. That is, it decreases gastric secretions, so it reduces the production of acid, it reduces the production of pepsin, and it also reduces mucus secretions. The other hormone is called cholecystokinin, or CCK, and CCK is also generated in the intestinal epithelium, but it travels to the stomach where it inhibits stomach emptying. That is, it slows down the muscular contractions of the stomach. And so in a nutshell, the intestinal phase is a way to basically put the brakes on gastric digestion to prevent the intestine from becoming overloaded from food that's being processed by the stomach. So we already talked about gastric emptying, but just to give a definition for it, gastric emptying is the periodic release of chyme from the stomach to the small intestine, and it's regulated by both nervous and endocrine signals. So things that help to promote gastric emptying include gastric distension, that is the stretching of the stomach wall by copious amounts of food. Presence of proteins will also stimulate gastric emptying, as will alcohol and caffeine. If you've ever sat around studying for a test, eating lots of pizza, and maybe drinking a lot of coffee, you know that things tend to move through very quickly with both alcohol, caffeine, and sometimes even nicotine. These tend to stimulate gastrin secretion, and gastrin causes increased production of mucus, increased production of pepsin, and also increased production of acid. These stimuli will also result in increased numbers of parasympathetic nerve impulses, and all of this will have the net effect of increasing the stomach motility and increasing the rate of gastric emptying. Now on to the factors that inhibit gastric emptying. The first of these is duodenal distension. Remember, the duodenum was the first loop of the small intestine. And as food passes through the pyloric valve in the duodenum can become distended or stretched with a lot of food. If this happens, we're going to inhibit gastric emptying. The other thing that can inhibit gastric emptying is the presence of those fatty acids or glucose in the intestine. And so if you eat a very, very fat-rich meal, it's going to have a lot of fatty acids in there, and these fatty acids are going to bind to the intestinal epithelium, and they're going to cause increased secretion of cholecystokinin and also decreased parasympathetic nerve impulses. And this is going to have the net effect at reducing stomach motility. The other thing that it's going to cause is going to cause production of secretin. And remember, secretin has the effect of reducing secretion of the enzymes within the stomach. So the net effect of lots of fats in the intestine or a large degree of duodenal distension is that we slow down what the stomach is doing so that the intestine will have time to process this food. So one thing you might have gleaned from the previous slides is that our rate of digestion depends on what types of foods that we're eating. Some foods digest faster than others. Carbohydrates, for example, have an extremely fast digestive time. If we're just talking about eating, let's say, glucose or eating grains that don't have any fat or protein in them, they can be digested very, very quickly, and you can expect them to move through your digestive system in only a few hours. Proteins, on the other hand, take a little bit longer to digest than carbohydrates do. And so if you eat a protein-rich diet, let's say you eat a steak, it's going to take several hours to digest, whereas carbohydrates can be digested in maybe 2 to 3 hours. And finally, fats or lipids have the slowest digestion time. Lipids take a long time to digest, and as a result, our gastric emptying time is going to be greatly extended. And so we can use this information to our advantage. For example, if you're going to run a marathon tomorrow. If you're running a marathon tomorrow morning, what is your diet going to be like tonight? Well, you're probably going to eat a high carbohydrate diet. There's lots of energy in there, but it also digests very quickly. You could probably have a little bit of protein in there, maybe a little bit of lean meat, let's say some chicken, but either way, this will ensure that this diet has completely been processed by your digestive tract before you get up to run the race the next morning. On the other hand, think about the fact that you're going to get up in the morning and eat breakfast and maybe not eat all day long. What kind of foods do you want to eat? Well, if you don't want to feel hungry, you probably want to eat foods that are high in fat and also high in protein, because these take the longest to digest and are with us for the longest. And we use this information to our advantage, for example, in planning the Atkins diet. The Atkins diet is a very protein and fat-rich diet which has minimal to no carbohydrate in it. And the reason that it works is the increased protein and increased fat actually leaves us feeling fuller for longer, and so we don't tend to snack as much as we would tend to if we were on an all-carbohydrate diet. Now, I want to be clear, I'm not advocating by any stretch of the means that anybody go on the Atkins diet. There's a lot of reasons why you don't want to eat a high-fat, high-protein diet for a very long period of time, and we'll talk about those in the next chapter in nutrition. So we're going to take a break from the GI organs at this point to take a look at some of the accessory organs that are located around the stomach. These include the pancreas, the liver, and the gallbladder. Remember, the accessory organs don't actually have food moving through them, but they do secrete enzymes or other substances which will go into the digestive tract, or they absorb nutrients that come from the digestive tract. So the first of these organs we're going to talk about is the pancreas. If you remember back to the endocrine system, we said the pancreas was an essential endocrine organ and that it produced both insulin and glucagon. These were hormones that helped to regulate our blood sugar levels. Remember that insulin tended to bring the blood sugar down, whereas glucagon tended to bring blood sugar back up. So these are endocrine functions of the pancreas, but the pancreas also has a very important exocrine function. Remember, exocrine glands have ducts which lead to a body surface or opening. And so the exocrine functions of the pancreas include the production of things like bicarbonate, as well as the production of different enzymes. And we'll talk about each of these in just a few minutes. So first, let's go talk about the anatomy of the pancreas. The pancreas, of course, has a head which is located proximal to the duodenum or small intestine. The body is the long part of the pancreas, and finally the tail is the part that extends away from the duodenum. Within the pancreas, there's a large pancreatic duct, which is a conduit through which the digestive enzymes and bicarbonate ions can exit the pancreas and enter the duodenum. Some of these secretions will enter the duodenum through an accessory duct. However, most of the pancreatic secretions will in fact enter the duodenum through something called the hepatopancreatic ampulla. And the word hepato means liver, and pancreatic, of course, means pancreas. And so this is an area where both secretions of the pancreas and also the gallbladder enter the small intestine or duodenum. So now let's take a look at the exocrine or digestive secretions of the pancreas. I already said that sodium bicarbonate was an important component produced by the pancreas. And sodium bicarbonate is a base. Think about where you have sodium bicarbonate in your house. Think about that yellow box of Arm & Hammer baking soda. And baking soda is a weak base. And weak bases help to buffer the pH. That is, they help to neutralize the acid that's coming from our stomach and bring it back up to a more acceptable pH. This is important because in the stomach we had lots of alkaline mucus that helped to protect the underlying tissue layers. We don't have so much of this mucus in the small intestine because that would inhibit absorption, which is one of the main functions of the intestine. So in order that we don't accidentally digest all of our intestinal cells, our pancreas will inject a little bit of sodium bicarbonate into the duodenum, which will bring the pH back up from, let's say, a pH of 2, which was in the stomach, all the way to a pH of maybe 5 or 6, which is what we'll have in the small intestine. Another secretion of the pancreas is pancreatic amylase. This helps to break down starch or polysaccharides into monosaccharides. Remember, we already had an amylase digesting enzyme within the mouth called salivary amylase, so why do we need a new one? Well, the reason is that amylase enzyme probably got broken down inside the stomach. Remember, the stomach had a very acidic pH, around 2 to 3, and chances are it would inactivate any of the salivary amylase that was swallowed. And so that salivary amylase is no longer around to help us digest carbohydrates. So here we produce a new amylase, pancreatic amylase, which will continue carbohydrate digestion. The pancreas also produces several protein-digesting enzymes, including trypsin, chymotrypsin, and carboxypeptidase. The one that I want you to know is trypsin. That's the primary protein-digesting enzyme manufactured inside the pancreas. And again, you might say, well, why do we need more protein-digesting enzymes? Didn't that happen inside the stomach? Well, it did, but pepsin, the enzyme that works in the stomach, worked well at a pH of 2 to 3. But now we're going to be at a pH of 5 to 6. And so pepsin is probably not going to work very well inside the intestine. So this is why we need to make new and different types of protein-digesting enzymes. And remember, the important one here is trypsin. Now, the next enzyme that's produced by the pancreas is something called pancreatic lipase. And lipase, as the name implies, is an enzyme that breaks down fats or triglycerides. We already talked about a couple different lipases produced both in the mouth and also in the stomach, but pancreatic lipase is actually the first lipase that really does a lot of work. And so if I ask you on an exam, where does fat digestion begin, the correct answer is going to be within the small intestine, because this is where the pancreatic lipase becomes activated and will begin to break down triglycerides into monoglycerides and free fatty acids. And finally, the last secretion of our pancreas is ribonuclease and deoxyribonuclease. These are enzymes that help to break down the DNA and RNA in the food that we eat. Remember that all the food that you ate was at one time alive. It was either a plant or it was an animal. And all plants and animals have DNA, so it's essential that we chop up their DNA so that we can reuse the nucleic acids for our own body. So the most important digestive enzyme produced by the pancreas has got to be trypsin. And trypsin was an enzyme that's going to help us to catabolize protein into amino acids. Just like pepsin, trypsin is in fact secreted in an inactive form called trypsinogen. Once trypsinogen enters the small intestine, it will be activated by an enzyme called enterokinase. Enterokinase is a membrane-bound enzyme found within the intestinal epithelium. This will help to convert trypsinogen, which is inactive, into trypsin, which is the active form of the protein-digesting enzyme secreted by the pancreas. Now, just like we saw in the stomach, production of pancreatic enzymes and bicarbonate, collectively called pancreatic juice, is regulated by both parasympathetic nerve impulses as well as endocrine signals from the intestine. In general, parasympathetic nerve impulses from the vagus nerve will stimulate moderate secretion of all of the digestive products of the pancreas. That is, there will be some bicarbonate, there will be some trypsin, and there will be some other enzymes in there as well. On the other hand, secretion of intestinally produced hormones will result in secretion of specific enzymes or secretory products. For example, an increase in secretin levels will cause the pancreas to secrete pancreatic juice, which is rich in bicarbonate ions, whereas secretion of CCK will cause the pancreas to secrete pancreatic juice that is rich in digestive enzymes. In this way, the pancreas can tailor the composition of the pancreatic juices to better match the type and quantity of food that is arriving from the duodenum from the stomach.